Hello everyone, welcome to my presentation. My name is Frank Fields. Today I'll be presenting on work that was done over the last several years at UC San Diego. This work is focused on demonstrating the capabilities of a new isolate of Chlamydomonas and the potential to produce a recombinant protein from it in an alkaline raceway pond. All right, I'd first like to give an overview of our strain production pipeline that we've developed at UCSD over the last 10 years. In the Mayfield lab here, we are usually focused on working with Chlamydomonas reinhardti, producing recombinant proteins to try and increase product value in the biomass that we can grow. Our tools typically have been limited to just synthetic biology, but over the last several years, we've developed additional screening methods and breeding tools that we can use to increase the protein titers of our host strain here. So this pipeline here just shows a typical overview of what we're generally doing. We're engineering strains with recombinant protein. Typically, they have a GFP component or some other fluorescent tag. The strains are then put through antibiotic selection and then onto or through cell sorting and pulling out the highest expressing individuals, which will then be characterized. And then we can use either UV mutagenesis or actual sexual breeding with Chlamydomonas to generate diversity within our cell line, recover those, and again, screen them and just turn the crank on this pipeline here until we get a strain that we're happy with. So of all the strain improvement tools that we have available at our disposal, the one that I want to highlight the most is the capability of doing sexual breeding or mating with your cell lines. And this is something that is very rare in microalgae, and it's one of the reasons that we really like working with species inside the Chlamydomonas genus because it's easy to do in a laboratory setting, getting good consistent results, and it provides a really unique tool that allows you to greatly increase the diversity found in your progeny and rapidly advance the expression of a specific target or generate adaptions through directed evolution. So this is something that we're always looking for when we are trying to isolate new strains. We want to find out, do they breed with our current library, and how can we expand the options that we have for doing these crosses and introducing genetic variability into our gene pool. So here's an example of some work that we did, putting a strain through our peak process pipeline here. This is a strain of Chlamydomonas reinhardti that was engineered with green fluorescent protein in the chloroplast. And the goal of this project was to figure out how to increase the expression of that protein using just breeding and mutagenesis and not using any other synthetic tools. So in this bar graph here, you'll see the expression of green fluorescent protein after different improvement strategies. The first is just the synthetic biology there, set to a relevant score of 1. And if we cross the recombinant strains with wild type strains, we got an increase between 6 and 7-fold, which was about the same as if we did just UV mutagenesis. And following each of those, we are putting these through cell sorters again and pulling out the highest expressors. However, what we learned is that if you stack these together, as it's traditionally done in a lot of industrial microbiology, doing mutagenesis first, screening them, and then doing breeding, and then screening them again, we got a 15-fold increase in our GFP expression, and this only took 17 weeks to do. So if we were to do this with synthetic tools, it would take significantly longer, and we've never been able to get results this high regardless. So this really set us up to go out and try and isolate strains that had characteristics that we were looking for, but were also capable of breeding, so we can put them through this process and try and replicate these results in a more productive strain with a variety of different proteins. So as part of this project to expand our strain library, we set out on a isolation trip to Southern California around the San Diego coastal areas and the Imperial Valley. primarily focused on isolating strains of Chlamydomonas that would be advantageous for outdoor cultivation. And then we were going to test and see if they were capable of breeding with our current strain library. So we had a stroke of luck and actually found this strain in a flower pot on UCSD's campus, of all places. And when we isolated it and brought it back into the lab, it was actively breeding already. So we were able to immediately isolate two breeding types, a plus strain and a minus strain. When we tested it with our current vectors for Reinhardti, they integrated perfectly and it expressed GFP, shown here on this Western, without any modification to the vectors. And then when we screened it for growth characteristics, we found that it was incredibly robust, tolerated a wide range of pH from 3 all the way up to 11, as well as tolerating very high temperatures up to 45 degrees Celsius. So this strain was deposited at the Chlamydomonas collection, CC 5697, and the partnering mating strain is available there as well. All right, so here is one of our first cultivation efforts with this Chlamydomonas strain. And I want to clarify, we did do 18S sequencing, and it is not Reinhardti. When you put it on a tree, it falls into an uncharacterized group. But we cultivated both Reinhardti and this new 5697 strain in these raceway ponds at our greenhouse at UCSD. And what we found was what typically happens with Reinhardti is that after about a week or so, the pond gets contaminated with bacteria, the cells get covered, the pond crashes and dies and looks like that. This other strain was grown at the exact same time, right next to it, and there was bacteria in the pond, but it did not adhere to the cells like it did with Reinhardti. The pond did not crash. It grew very well, as you can see by these pictures here. So we were very happy to see that, what we observed in the laboratory starts holding up outdoors as we grow this in an open pond. We also did a lipid analysis of 5697 in both replete and deplete conditions. Nothing too surprising there. Looks like pretty much every other green algae, getting up to about 12, maybe 13% when we depleted it of nitrogen in these ponds. All right, so now that we confirmed that this is certainly a strain of interest for us, we went and put it through the same protocol that we've been using for our Reinhardti model. We engineered this with a nuclear vector that is expressing GFP and targeting it to the chloroplast, screened on double selection, put through a flow cytometer, and pulled out the highest expressing individuals and confirmed them with both microscopy and Western blot. We also put out another paper detailing this method of rapid transformation and screening to get high expressors around this time. Details are there for those of you that are interested. So now that we had both a wild type and recombinant variant of this strain, we wanted to do a side-by-side outdoor cultivation test to understand how the two strains compared and if any productivity would be lost while we're producing a recombinant protein. Since this strain did tolerate such high pHs, we actually elected to do this trial without any supplemental CO2. So the ponds were alkaline in nature, and the pH was above 9 up to 11 the entire time during this cultivation trial. You can see the temperature fluctuations there on the top right, and underneath that is the O2 saturation, showing you essentially the photosynthetic rate of each of the ponds there. They were very similar in nature. The bottom left shows your productivity here. On the left side of the axis is the volumetric density in grams per liter. On the right side is the areal density, and every dot there is a single day, a 24-hour time point. So we did essentially 5 knockbacks, or 4 knockbacks and 5 growouts of these strains over the course of a month. We let them grow up to a certain density, and would do essentially a partial harvest. About half to 75% of the pond was harvested and then refilled with fresh media, but again, still no gaseous CO2 was being put into the ponds. And overall, when we looked at the productivity, they were really similar to each other. We didn't see any significant difference between the productivity of our wild type and the recombinant strain. We got about 8 grams per meter squared per day, which is not a very high number, but considering that this is a newer strain and we're not using CO2, we found it to be pretty acceptable for this experiment. And then finally, the bottom graph there on the bottom right shows the GFP fluorescence over time. So the orange dots there obviously showing you the recombinant strain, where our GFP is present in all of our samples. It does trend downwards over time, which is as expected when you continuously cultivate a strain like that. You expect to lose some expression over time. However, it did not ever drop down to the wild type levels. Okay, so I thought I would also take this opportunity just to showcase a little bit more of what UCSD had going on during this experiment. So on the left there, you can see our 3 ponds fully set up, growing the 5697 strain. And we grew them up to saturation, put it through a Zobi harvester that we had gotten from Global Algae as a partner on this project. The strain harvested very easily, going through the membrane filtration systems. The paste or the concentrate that came out the other side was put through a continuous flow centrifuge to make an algal paste that shown there in that bottom picture. Very non-odorous, very easy strain to work with and deal with. So overall, we're very happy with this new isolate. It seems to have all the tools that you could possibly ask for in an algal strain. And we really look forward to seeing what we can do with it in the future. I'd like to acknowledge all the UCSD contributors listed here as contributing significantly to the work shown here today. Thanks to UCSD for their facilities and support, Global Algae for their partnering on this project and providing some equipment. And then funding for this was provided by the U.S. Department of Energy through the 2 grants listed below there. So thank you so much for coming to the talk, and hope you enjoyed it. Thanks.